Jai Hind everyone, I am Pankaj Kumar and today we are going to read about the topic plant tissue culture. This is very important topic. From this topic GAT B will ask 2 to 3 questions and I will tell you in which subtopic they will ask more questions. So the topic is the chapter, you can see it is under the recombinant DNA technology. You can consider with plant tissue culture. Plant tissue culture, okay. In this plant tissue culture, we will take any cell, any organ, any cell, tissue, or organ of root, leaves, shoot, or other parts. Okay. So, so what we will do as of now, we know we have to take. This is a plant. Either we can take a root cell or shoot cell and leaves. So which kind of cell we should take? Okay, so in plant tissue culture we can take any kind of tissue, cell or organ of a plant, but we will prefer the meristem. It depends also on the types of tissue culture. Okay, so we will take the root, shoot or leaves cells and then we will grow in a medium. So suppose this is the any cell. Now this is any plant cell. Now we require what? We require some supplement so we can grow it in vitro condition. What is in vitro condition? In vitro condition we will grow a plant. Will grow in a plant in outside the body. Okay, in a tube or in a experimental way. Okay, outside the body we can say. So we will add some supplement. What we require? Supplement required. In supplement, what we will give? We will give the nutrients. Okay, and nutrients is of two type. We can say inorganic and organic. Inorganic, you will see in inorganic type. The first is what? The first is micronutrients. Okay, micronutrients. In micronutrients, inorganic salts are what? Can you guess? So iron, zinc, copper, manganese, iron, zinc, copper, manganese, and all these things. Okay, so these are the micronutrients which we have to provide. And then second is what? Macronutrients. Macronutrients are what? Nitrogen, phosphorus, sulfur, potassium. Okay, these all are what? Macronutrients. Apart from that, we required what? We required organic compound also. So which kind of organic compound we required? We required a vitamins. Usually we required a vitamins, and these vitamins are what? Vitamins are like thiamine, niacin, folic acid. Okay, this is vitamin B1. This is B1. Okay. Thiamine, niacin, folic acid, and in organic only we will write the sugar. Required sugar. Okay. So to maintain an environment of this cell like a plant, okay, to maintain an environment of this cell like a plant, we have to maintain the pH. So pH will help in uptaking of all these things, thiamine, niacin, in macro and micronutrients inside the cells. If the pH will be not maintained, then what will be the different consequences? Maybe cell will die, maybe cell will survive, but the growth will be not occur. So these are the different things. So second thing what we will see, we added a supplement. Now we have to maintain. Then we have to what? We have to maintain pH. How much pH we have to maintain? So for most of the tissue cultures, we have to maintain around 5.7 pH. Okay. How it will help? The pH will help in uptaking of these nutrients. Okay, it will help in uptaking of uptaking of nutrients. Maintain maintains soluble maintains soluble what maintains soluble salts. Okay, and then this pH is very important for the enzymatic reactions. Okay, it will promote enzymatic reactions. Okay, if the pH will be not proper, the enzymatic reaction cannot be occur. Okay, enzymatic reactions we can write. Hope so. Here, up to here you understood. Okay, up to here we understood. Okay, now we will see. Okay, so now we will see that enzymatic reactions are which kind of enzymatic reactions we are not interested. We are interested in how pH will be helping in the helps in the tissue growth. Okay, we took at the cell, we added a supplement, then we added a pH, we maintained the pH. Got it? Now we'll see. So till now we've seen supplements and pH. Can you guess anything else which is required for the plant growth? What are that important thing for our cells? Our also it is required for the growth. That is hormones. So hormones are very plays a very important roles in the growth of the plants. So what are the hormones are there? So the first hormone is auxin, and this auxin how it will help in the formation of tissue. Okay, I will tell you the first one by one. We'll see. Okay, so auxin is at high concentration. At high concentration, this is concentration. The adventitious root will form. Okay, at low concentration, the adventitious shoot will form. Okay, and it will inhibit axillary buds formation. Okay, axillary bud formation. Okay, and it will help in one, two, three, fourth somatic embryo formation. Okay, somatic formation. So these are the roles uh, which will be presented by the auxins. Apart from this, some more functions are there. Auxins functions are there, but we are focused on the tissue culture, so we consider only the tissue culture related functions. Second is what? The second is we can write cytokinin. In cytokinins, what is the important thing? In cytokinins, if you will see at high concentration, cytokinin at high concentration, the adventitious shoot will form. Okay, and then it will help in uh, okay, it will inhibit root formation. Okay, and then it will inhibit the leaf senescence. It will inhibit leaves to inhibit leaf senescence. These are the role of the cytokinin. If you see, the oxygen is discovered by afferents, and cytokinins like what are the cytokinins? Like anatins, and these all discovered by the described by Scugenmiller. Okay, so these are what hormones which is important for the plant tissue culture. Now we will see another hormone. What it is? That is our gibberellin. Okay. So third is our gibberellin. In gibberellin, what you will see, gibberellin is very important hormones. It will improve the, it will help in the bolting. It will improve the length of the stems. Okay, increase the internode sizes, lengths. Okay, so gibberellin here we will see it will inhibit adventitious root formation. Okay, in in all in cytokinin it will inhibit the root formation complete, but here it will inhibit adventitious root formation. Okay, then it will help in second, it will help in the seed germination. Okay, it will help in seed germination, but here we are not using as a seed in tissue culture, but we can use in the different type of tissue culture. Got it? Seed germination, and the most important thing, internode elongation. So anyhow, it will be very important for the growth of the shoot. Got it? Second thing is what? After gibberellin, we have what? We will see ethylene. Okay, so in ethylene we will see the function of ethylene. What it is happening? It will help in the ripening of fruits. It will promote senescence of leaves, old leaves. Okay, of old leaves. Usually, it will help in the ripening of the fruits, senescence of the old leaves, and then next is what? So next will be the uh, jasmonic acid and the polyamines. Okay. So it is fourth, fifth polyamines. So polyamines are the very important hormones because it is helping in the somatic embryogenesis and somatic and promotion of adventitious root. So it will help in somatic embryogenesis. So taking any somatic cells other than the reproductive cells and forming a embryo from that cells. Okay, that is called somatic embryogenesis, and then it will help in the promotion of adventitious root. Promotion of adventitious root. Now then you can see the jasmonic acid. Okay, which is the next hormone, that is the jasmonic acid. So jasmonic acid will help in the growth of what? This meristem. Okay, growth of meristems. So it will increase the meristem size and the numbers, and then after that it will help in the formation of bulbs and mm, birds. Okay, bulbs and birds. So these all hormones are required to grow a plant, but abscisic acid is not required here. Okay, but that is a what it is? It is abscisic acid is a stress hormone we can call. So these are all the important hormones which we have to remember and their functions. I told the most important functions for the tissue culture. Some more functions are there. Okay. Now we'll see one important term that is very important for the growing of a cell. So this is a plant, and I want to grow a new plant from this plant. What we can do, we can wait for the seed. Once the fertilization will occur, then seed will be formed. That wait we cannot do. Why we want tissue culture? Because it's very fast, and you can grow numbers of tissues, okay, numbers of plants within a few time, within a less time, okay, as it is occurring in the TNPL. Okay, so now I there is a two sides. I took A side and B side. From which side it will be better? So growing a tissue or a new plant by a tissue culture, it's better to take a meristematic cells. So this is a tip. Here more meristematic tissue cells will be there. So we can take A side better. But what if I will take from the B side? So here no meristematic tissue are there. So these cells, the cells which are present at the B side, that are the differentiated cells. What are it is? Because they already attain their proper function. Okay, they attain their proper function. Now they are not dividing cells, not dividing cells. Got it? But at the cells which are present at the A side, that are meristematic, they are dividing cells. That is called that is called meristematic tissue, and they will attain a proper function. Okay. So B is all non-dividing cells, B side not dividing cells. Then if we are taking the B cells, then we have to induce this cell by cytokinesis to divide in new cells. So this is a non-dividing cells. Okay, non-dividing non-dividing cells. And you once you will add a cytokinin, then it will start dividing, and from non-dividing cell it become what? Dividing cells. So that is called what? De-differentiation. What it is called? De-differentiation. What it is called? It's called de-differentiation. You can write it here. De-differentiation. So first what we have to do? We have to attain the de-differentiation. Then only we can go for the further movement. So again, after being a de-differentiated cell, it will be transformed into a proper defined cell. Now it is a defined cell. Defined cell means what? I want to tell you, defined cell means it is attain their own function. Like so from stem it can form a xylem, phloem, cortex, hypodermis, pith, still, like any part of this tissues. Okay. So this is defined cell. They attain their function. So that reversing from de-differentiation, it is called re-differentiation. The process is called re-differentiation. Okay. Yes. Re-differentiation. Okay. Got it. So this is the one more term is that differentiated cells. Like though now it attain, now this cells which is non-dividing, that was called as a differentiated cell. Okay. So hope so you understood de-differentiation, re-differentiation. We have to induce a cell to divide it again. That is called de-differentiation. And once the cell again it will regain the defined function, then it is called re-differentiation. Or one more term, that is a differentiated cell, means cells which is already not dividing and they attain their function. Hope you understood these things. Now we'll see. Now we'll see. Now we'll see the next topic, which is now we'll see the next topic, which is about the sterilization. Okay, I will tell you the sterilization. Before putting all these things in a tissue culture, we have to add, we have to sterilize that thing. Okay, so how we will sterilize? So sterilization will be done, cannot be done by the heating, because these hormones are very sensitive to the heat. So if you put the hormones along with the heating, then it may be degraded. Okay, so we will not use the heating process. We are not going to use the autoclaving process for this media. Okay, or for this vitamins. So what we will do? We will use a sterilization method. Digestion method. So for that we have a, we will take 1 to 2% sodium plus 9 to 10%, 9 to 10% calcium hypochlorite, and then 70% ethanol. We will take all these things and then we will put into the media. We will pass the we will take the tissue, cell, stem part, anything. Then we will wash it with this, and then it will be sterilized. Like that, we will use for the sterilization. Okay, so there will be different stages of sterilization. We can go through that, but up to here for your level for the GAT B and IITJAM, this is sufficient. So now there are different type of tissue cultures: Callus culture, cell suspension culture, uh, embryogenesis, then ovule culture. Okay, so somatic cultures. So there are different type of cultures. Okay, so we will see the Callus culture. Got it? So we are continuing the Callus culture and the embryo culture. In this video, we will end up with this two cultures type. In next videos, we will continue with the remaining cultures. Got it? In Callus culture, what we have to understand, that is a callus we will take from the plant tissue, and from plant then we will grow as usual with the supplements and the pH and the hormones. So what is callus? Callus is an undifferentiated, unorganized mass of the cells. It is not differentiated cells. So we can say it is a meristematic tissue. Okay. So if it is a meristematic tissue, then it will grow fast and it will be having more dividing capability. So the three stages of the callus is what? Induction of the cell division. We will induce the cell division by the what? By the help of auxin and cytokinin. So here you just mentioned one very important thing from which they will ask the questions most of the time. That is, if auxin and cytokinin is equal, then cell will divide usually. Okay, cell division occur. For differentiation, this is very important. What is the very important? For differentiation, this is very important. What I am going to tell you. So cytokinin by auxin. If it is very high, then it will give. If it is very high, it is high, then it will give uh, shoot. Okay. Or I think root. Wait. If it is very high, it will give you the root formation. And if it is if auxin by cytokinin kinin, so if auxin cytokinin ratio is very high, it will give the root. Okay, it will give root. So we have to change it here. It was correct. It will give the shoot. This is very important because they ask in different examination also. They ask also in the gate, in IITJAM and then GAT B also. Okay, so they can ask in the CUT, UPG also. But this is important term. You just mention it. So which kind of cells it can be grown? That is a totipotency cells. Okay. So the totipotency cells is undifferentiated cells which can regain the division capability. That is called totipotency. So human cells, all cells are not totipotencies. Okay, but you can see plant cells. You can take a leaf. You can take a intercalary meristem, or you can take any cortex cells. You can take uh, any branch cell, okay, any root cell, then it can de-differentiate, okay, that have a capability to regain a cell division or to regain a de-differentiation capability, that is called totipotency. Our human cells or animal cells are irreversible or de-differentiated, okay, means it cannot regain a cell division. If I will take my skin cells here, if I will grow, it will not going to divide into new cells, okay. But if I will take a cells from the tissue plant stem, it I will grow, then it will divide. That is called totipotency. Got it? Now other terms are there in terms of the human being, like in terms of animals, like multipotency, pluripotency. I'm not going to discuss here, but we will see. If you want, I will tell you in the comment box. So, induction of the cell division. We have to Callus culture will be done by the induction of cell division to de-differentiation. After de-differentiation, it will attain a proper cell line or proper function. Then the cell division will be ceases, and after de-differentiation, cell division will be ceases. Now then we can see embryo culture. Embryo culture is what? Isolation of the mature or immature zygote embryo aseptically. With proper sterilization, we have to isolate a any embryo. So it is very tough to isolate an embryo. So what we will do, we will take a complete ovule or complete ovary. Then we will call it as a ovule or ovary culture. Okay. So embryo directly we will isolate. After isolating, then we will put into the supplement, and then it will grow. So it depends. If embryo is mature, it will grow faster. That is concluded by Dietrich. Okay. If embryo is immature, then it will take time. So the first scientist Heng, who cultured a matured embryo in presence of salt and sugar supplement, Dietrich cultured both immature and mature embryo, and he found that among both, matured embryo will grow very fast. But advantage is why we want an embryo, because sometimes the fertilization will be occur, or but the seed will be not formed. Like in case of hybridization, in case of apomixis. Okay, so in case of hybridization, that we can take an embryo and we can induce a viable seed from inviable seeds. Okay, so in inviable embryo which is not able to grow matured into a seed, then we will try to induce that type of embryo into a complete proper seed. Okay, so for overcoming embryo abortion in the self incompatibility, these are the advantage why we are using embryo culture. And then overcoming of seed dormancy and self sterility, and embryo rescue and breeding cycle, decrease the breeding cycle. Embryo rescue means what? Sometimes the embryo formed, but there is a very less or no endosperm. In that case, we can use this method. Okay, and obviously it will decrease the breeding cycle. So till now, hope you understood. In description there will be link for the biology app. There you can go and practice your questions regarding these topics, and you can attend the live classes by typing on the biology app. Thanks for watching. We will meet in the next video. Till that, Jai Hind.